Hello children, let us continue with respiration in plants, the second lecture in the series. In the first, we have learned about what is glycolysis. Glycolysis is the splitting of glucose molecules to yield two pyruvate molecules or two pyruvic acid molecules, and in that process there was yield of two ATP and two NADH. We have already seen those in our first lecture. Now let us continue what happens after glycolysis in respiration. Now, after glycolysis, you will see that the glycolysis, which is splitting of glucose to yield two pyruvic acid molecules. Now, those two pyruvic acid molecules, they are present in the cytoplasm of the cells. Okay, please remember glycolysis takes place in cytoplasm of the cells. Now, depending upon whether the cell does not have oxygen or has oxygen, absence or presence of oxygen, accordingly the rest of the reactions will proceed. If there is absence of oxygen, then the yield will be either lactic acid or ethanol. It will depend upon whether which type of enzyme is present and where is the reaction taking place. That means the cell belongs to which type of organism. And if oxygen is present, sufficient quantities, then you will get the yield of carbon dioxide and a very high number of ATP molecules. So accordingly, we call the first, that is in absence of oxygen, we call it as fermentation, and in presence of oxygen, it goes for Krebs cycle. Okay, so we will talk about all these details in this present lecture. Now, after glycolysis, this is glycolysis, glucose converted to pyruvic acid molecules. Okay, converted to pyruvic acid molecules, and herein what is happening here, there is yield of 2 NADH and 2 ATP. Also water is yielded. Now these pyruvic acid molecules, assuming that there is no more oxygen available, no oxygen is available in that particular cell. Okay, in that particular cell, let us assume that there is no oxygen available. Then what will happen? If lactate dehydrogenase, the enzyme lactate dehydrogenase is present, then it will get converted to two lactic acid molecules. Okay, two lactic acid molecules, which usually is seen in some bacteria as also in mammalian muscle cells, even in us, in humans. Okay, it happens. Like suppose you are exercising, you are exerting any part of your body, you are carrying some load, you are running a lot, you had to run a lot. Okay, so a lot of energy is required to that particular organ. Now what will happen? Initially that yield will be there with the oxygen that is available in your body. A point may come, demand of energy is more but the supply of oxygen is less. Then in that case, deficiency of oxygen occurs in that particular organ. So there what will happen? Glycolysis. After glycolysis, the pyruvic acid will get converted to lactic acid. There will be yield of energy, so you will be getting energy and that particular organ will get energy, but there is no proper respiration taking place. It goes for lactic acid fermentation, and that will result in accumulation of lactic acid that may sometimes even create some problem in that particular organ. But whatever be it, in absence of oxygen, lactic acid formation takes place. And in microorganisms like yeast, what will happen? If there is oxygen is absent, then the pyruvic acid molecules will get converted to ethanol and carbon dioxide. Also some minimal amount of energy will be yielded. That is possible in presence of alcohol dehydrogenase. Okay, alcohol dehydrogenase, the enzyme, will be responsible for this particular reaction. So this is known as fermentation. In absence of oxygen, the fate of pyruvic acid, the conversion of pyruvic acid to lactic acid and ethanol occurs. Now we go for aerobic respiration. Aerobic respiration is the condition, this is normally which takes place in plants as well as in animals. Now what happens in aerobic respiration? There is supply of oxygen. Sufficient supply of oxygen is present when the pyruvic acid molecules formed after glycolysis will go further, break down further reactions to finally yield carbon dioxide and a high amount of energy. That is called as aerobic respiration. Now what happens? Now the pyruvic acid molecules that is formed in the cytoplasm during glycolysis will enter into the mitochondria. Now if you can recall, this is the structure of mitochondria which we had already done in the chapter on cell. Now mitochondria, it is a double membrane structure. This is an open, like the mitochondria has been opened up to show us the internal structure. Double membrane, okay, this brown and this grayish structure, double membrane structure, this bluish, okay, it is filled with a fluid like structure called as matrix. The inner membrane is highly folded, which are known as cristae. Now there is also a space in between the outer and inner membrane. This space is also of great significance during respiration. So what will happen? Pyruvic acid, which is in the cytoplasm outside the mitochondria, will now enter into the matrix of mitochondria. Okay, it enters into the matrix of mitochondria. Then after that, it follows two steps. Okay, two steps. The first is Krebs cycle. Okay, Krebs cycle, which will occur here, and in it there will be during Krebs cycle, it's a cyclic reaction, is a cyclic reaction whereby what will happen, the products of pyruvic acid, okay, the pyruvic acid, of course there is one more link step, we'll talk about it. So it will go through a cyclic form of reaction and yielding NADH, okay, ATP molecules, NADH and FADH2. These are the molecules that will be formed during Krebs cycle that will further help in, or that will further go for oxidative phosphorylation. Okay, so these are the basic two steps in aerobic respiration: Krebs cycle and oxidative phosphorylation. In oxidative phosphorylation, what will happen? The NADH and FADH2, they will undergo oxidation reduction reactions upon this inner membrane and yield ATP. Okay, yield ATP. That is the final reaction when there will be production of energy. Now there is a link step of glycolysis and Krebs cycle. Okay, after glycolysis, when the pyruvic acid molecules have reached the matrix, then a reaction occurs which is known as pyruvate decarboxylation. From the term decarboxylation, you can make out removal of carbon. So there's a link step, very short step, which will occur here. After that, Krebs cycle starts. So here in the link reaction, what happens? PA is pyruvic acid molecules which gets converted to acetyl coenzyme A. It's a 2-carbon compound. It gets converted to acetyl coenzyme A, and in that process, during that process, what will happen? There will be yield of carbon dioxide molecule and also one NADH. Here coenzyme A enters into reaction, that means pyruvic acid plus coenzyme A plus NAD enters into the reaction and forms acetyl coenzyme A plus carbon dioxide, okay, and plus NADH. These are the yields. Now if you can recall, from one glucose molecule, two pyruvic acid molecules are yielded. Both the pyruvic acid molecules will undergo this link reaction, because finally what are we going to understand or learn? That what is the yield of energy from one glucose. So at every step you have to keep on reminding yourself that we started from one glucose molecule. It yielded two pyruvic acid molecules, so we write it into 2. Okay, twice this reaction will take place, one after another. Finally, there will be yield of 2 acetyl coenzyme A, 2 carbon dioxide, 2 NADH. Okay, 2 each. Now, just a brief outline so that you find it easier to follow again. This is the cell, mitochondria, cytoplasm. Okay, so here this is the cytoplasm where glycolysis took place. Here you see glycolysis has taken place, glucose getting converted to pyruvic acid. 2 arrows means 2 pyruvic acid molecules. There is yield of some amount of ATP. Then pyruvic acid enters, okay, into the matrix where this Krebs cycle or citric acid cycle takes place. there is yield of NADH. We will see this now, this particular cycle, and in between, here, in between, after it enters, this is the link step, where there is conversion of pyruvic acid to acetyl coenzyme A, which can start with Krebs cycle. So here, if you can recall, during glycolysis, NADH was formed. That also enters into mitochondria upon the inner membrane. Whatever NADH will be formed here, even this NADH will go for oxidative phosphorylation. If whatever will be yielded during Krebs cycle, that will also go for oxidative phosphorylation. That is here, this step, okay, oxidative phosphorylation, which is also called as electron transport and chemiosmosis. Okay, that will finally result into formation of ATP, very high amount of ATP. So the brief outline goes on in this manner. Let us see now citric acid cycle. Now here into 2, okay, it means twice, okay, because how many? 2 pyruvic acid gives 2 acetyl coenzyme A. Both of them will go for citric acid cycle. That will result in the reaction taking place twice. Then what happens first? Okay, this is acetyl coenzyme A, which is accepted by a compound oxaloacetic acid already present in the matrix of mitochondria. Okay, oxaloacetic acid, which is a 4-carbon compound, takes up this 2-carbon acetyl coenzyme A to form the citric acid or citrate, which is a 6-carbon compound. This citric acid, okay, further what happens? It undergoes changes. Of course, here enzymes are responsible to form isocitrate, okay, and that gets converted to alpha ketogluteric acid. During this step, there is yield of 1 carbon dioxide and 1 NADH per acetyl coenzyme A. So if we are considering 2 glucose, then here is the yield of 2 carbon dioxide, 2 NADH. Then alpha ketogluteric acid gets converted to, okay, succinyl coenzyme A. It gets converted to succinyl coenzyme A, and even this is a decarboxylation reaction where there is removal of 1 carbon dioxide molecule and NADH, formation of NADH. So here for 1 acetyl coenzyme A, you got 1 carbon dioxide, 1 NADH. For 2, we will get 2 each. So finally, so here what are we getting? 2 plus 2, 4 carbon dioxide, 2 plus 2, 4 NADH. Now succinyl coenzyme A will get converted to succinate. Okay, it gets converted to succinate. Now during this step, there is release of 1 GTP, guanosine triphosphate, similar to ATP, same energy molecule. So here 1 for 1 acetyl coenzyme A, it will be 2 for 2 acetyl coenzyme A. Then succinate gets converted to malate via fumarate. Okay, fumarate is an intermediate compound. It will get converted to malate or malic acid. These are 4 carbon compounds. Now you can see here citrate was 6 carbon. Why? 2 acetyl coenzyme A plus 4 carbon oxaloacetic acid combines to form 6 carbon compound. There is removal of 1 carbon dioxide, removal of 2nd carbon dioxide, 2 carbons removed. So succinyl coenzyme A becomes again 4 carbon compound. Succinate 4 carbon compound, malic acid 4 carbon compound. During this step, there is release of FADH2. Then finally, what happens? Malate gets converted to oxaloacetic acid again, which is 4 carbon compound. There is again a release of NADH. Okay, so now oxaloacetic acid gets regenerated. You can see. Why is it required? To accept acetyl coenzyme A. So this way, in a cyclic manner, the reaction occurs, and during this cyclic pathway, there is release of 2 carbon dioxide, 3 NADH, 1 FADH2, 1 ATP per acetyl coenzyme A. But for 1 glucose, what will be the yield? 4 carbon dioxide, 6 NADH, 2 FADH2 and 2 ATP, because we are multiplying by 2. Okay, so this is citric acid cycle or Krebs cycle. Now further, let us see here. Okay, so this is the glucose. Okay, glucose molecule undergoing glycolysis. Okay, I'm summing up till Krebs cycle here. So here what happens, this is glucose undergoing undergoing glycolysis, fructose diphosphate glyceraldehyde. If you remember, splitting occurs, and there is yield of 2 pyruvate from 1 glucose. Okay, so that pyruvate, okay, what will happen, it will form acetyl coenzyme A. Okay, it will form acetyl coenzyme A, that is the link step. During this, there is release of 1 carbon dioxide. So for per glucose, 2 carbon dioxide released here. Okay, then it undergoes Krebs cycle. This is tricarboxylic acid cycle or citric acid cycle, or you can also call Krebs cycle. Different names for the same reaction. So just now the reaction that we saw, so it goes on in a cyclic manner with a yield of NADH, FADH2, ATP molecules. Okay, now let us see what we have got till now. During glycolysis, glucose forming 2 pyruvate, okay, 2 carbon molecules. What is the yield? 2 NADH was released here, 1 and 2. 2 NADH, 2 ATP released. Okay, then transition reaction, that is the link step, 2 pyruvates getting converted to acetyl coenzyme A, okay, plus 2 carbon dioxide molecule. Okay, 2 carbon dioxide yielded here, also 2 NADH. Then during Krebs cycle, acetyl coenzyme A forms, releases 4 carbon dioxide. Here you can see okay, 4 carbon dioxide molecules are released. Then 6 NADH, as I had already shown to you. Okay, 6 NADH. Here they have not shown. Okay, here, okay, see here, 3 NADH, 1, 2 carbon dioxide, 1 ATP. Okay, so the numbers have been given here. So here 6 NADH, because this is into 2. Okay, 6 NADH, 2 FADH2 and 2 ATP. That is the yield. So total from glucose till carbon dioxide, okay, the total yield. Glucose has already got converted to carbon dioxide. That is the byproduct. We know during respiration there is release of carbon dioxide. So here the total yield will be 10 NADH, 2 FADH2 and 4 ATP. These are the direct energy molecules. But NADH and FADH2, these are the indirect forms of energy. This will again, now what will happen, these two molecules will undergo oxidative phosphorylation or through the electron transport chain to give ATP molecules. That we have to see in our third lecture. We will learn about it in our third lecture. Okay, thank you.